Okay, we're going to continue here in podcast 11. Here we go. We're continuing to talk about uh, genetics, and now we're considering some more complicated situations, and we're going to follow two traits at the same time. Okay, so in situations where we're following two traits at the same time, what we're talking about is that in any individual, they're going to have uh, two versions of the trait. One they got from mom on one chromosome, one they got from dad on another chromosome. Those two versions can be the same, like they're both yellow here in this individual, they're both green here in this individual, in which case that'd be the P generation or the parental generation, and these are true breeding. Or they could be different. So it could be one copy of yellow and one copy of green. When we're talking about two different traits, we're talking about genes that are on two different locations on chromosomes. Here I showed them on the same chromosome, but they can be on different chromosomes. And when the organism goes to make pollen or to make an egg for the next generation, they can only give one version of each gene to the egg or to the pollen. So that for these two individuals, there would be four possibilities. It could be yellow with wrinkled, it could be yellow with smooth, it could be green with smooth, or it could be green with wrinkled. Okay, so if this individual, which is yellow and smooth and true breeding parent, with a green and wrinkled true breeding parent, if they're mated together, all the F1 offspring will be yellow, dominant phenotype, and smooth, dominant phenotype. And they would be the F1 generation. And those F1 generations would all have the genotype like this. They would have one copy of the yellow gene, one copy of the green gene, one copy of the smooth gene, one copy of the wrinkled gene. They would be heterozygous at both loci. So in reality, those genes are on different chromosomes. I drew them on the same chromosome in the previous slide, but they're actually on different chromosomes. And so this individual would have a set of chromosomes that looks like this. One of the ones, say the one they got from mom, has a yellow gene on it. The one they got from dad has green on it. And the one they got from mom has a wrinkled gene on that spot. And the one they got from dad has a smooth gene from that spot. So when this individual, this heterozygous F1 individual, goes to make gametes, okay, eggs and pollen, we can set up the four combinations, and this is set up through meiosis, which is going to happen in a couple podcasts. I'll explain meiosis. And 25% of the eggs or pollen they make will have a big Y and a big S. 25% will have a big Y and a little s. 25% will have a little y and a little s. And 25% will have a little y and a big S. So this is what you're essentially doing when you're setting up a Punnett square, is figuring out from this parent what are the possible gametes. And this is the procedure that people have real hard time doing when they're setting up a Punnett square. Okay, so let's say these are our two parents. They're both heterozygous. They both have the same genotype. What kind of gametes can they make? Across the top of a Punnett square and across the side of the Punnett square are the gametes. So in the gametes, you have to have each gamete needs one version of each gene, one type of Y and one type of S, and that's it. Okay? So I'll start off with a big Y. Well, a big Y with what kind of S? Okay, I'll start and I'll say a big S. What's the other combination that has a big Y in it? Well, big Y with little s. Okay? And then I can also make a little y with a big S. And what's the other version of gamete I can make with little y? A little y with little s. Since this individual has the same genotype, they have the same kind of gametes. So this setting up the gametes is what people have the hardest time with. Okay? It's not that bad. You just got to remember this rule. Each gamete needs one version of each gene, of each type of gene. So there's two types of gene here. One is color, one is shape. I need one version of color and one version of shape for each of those genes. Then filling out the Punnett square is no biggie at all. So the individuals have two copies of everything, and they get it from the cross. So this egg crossed with that pollen, when this pollen grows into this egg, it contributes this big Y to that big Y, and the result is big Y, big Y. And this big S to that big S, and the result is big S, big S. If we go over here, little y and little y makes little y and little y, big S and big S makes big S, big S. Sometimes people get messed up because like here's a little y and a big Y and it's written big Y, little y. That's just a convention that capital letters go first in Punnett squares. Okay, so here's all the results of a dihybrid cross. What do we make sense of this? Well, okay, let's try to figure that out. Here's Mendel's data, and he shows that here's the number of P's that have dominant for both characteristics, dominant for color, but recessive for shape, recessive for color, but dominant for shape, and recessive for both color and shape. If we figure out the ratio for these, it's 9.8 to 3 to 3 to 1, 3.4 to 3.4 to 1. If we round these numbers, it comes out to 9 to 3 to 3 to 1. Now 9.8 you wouldn't say rounds to 9, but 9 to 3 to 3 to 1 is the theoretical ratio of what you would get from these combination of gametes. And as we saw previously, when we mess with sample size, you don't always get the theoretical ratio, and you have a better chance of getting that theoretical ratio with a bigger sample size. The more traits that you are going for, the bigger number of P's you got to count. So even though this is a big number of P's, it's not quite enough to come out to exactly 9 to 3 to 3 to 1, but that's the theoretical ratio. So there's 9 yellow, smooth, 3 wrinkled, yellow, 3 green, smooth, and 1 green, wrinkled. If we just do the ratio of yellow to green, it's still 3 to 1. That's about 400 to about 100. If we do the ratio of just round to wrinkled, that's about 400 to about 100. So if we just consider one trait, like color, that's still about 3 to 1. Or wrinkled, that's still, or smooth, that's still about 3 to 1, round over wrinkled. But the two traits together, it's 9 to 3 to 3 to 1 is the phenotype ratio. Okay. Once you get past a dihybrid cross, the Punnett squares get really big and not very practical to fill out. So if I go from 2 traits to 3 traits, now it's 64 squares you got to fill out. And considering that humans have somewhere north of 22,000 genes, that would be a whole lot of squares. And it gets to the point where it's not practical anymore. So you can use probability to figure out what is the likelihood of a certain trait showing up under simple dominance. And we'll talk about it's not always simple dominance at the end of this podcast. So probability goes from 0 to 1 or 0% to 100%. Probability of 0 means that an event will never happen. Probability of 1 means that it will always happen. Anything in between there gives you the probability of what will happen. So a coin flip, the chance of it landing on heads is 0.5 or 50%. So 50% of the time you flip a coin, you're likely to get heads. If you want to figure out the chance of two things happening, where this happens and that happens, and is the key word there, you can use probability. So if I'm rolling dice, there's 6 sides on a dice face. On one of those sides is the number 5, and the chance of me rolling a 5 is 1 out of 6. And the chance of me rolling a 4 or any other number is also 1 out of 6. But if I want to know what is the chance of me rolling a 5 and a 4 in that order, well, that's 1 out of 6 times 1 out of 6, or 1 out of 36 times I will roll. If I have 2 rolls, I will roll a 5 the first time and a 4 the second time. That's 1/6 times 1/6, or 1 out of 36 times. So use the multiplication rule whenever there's an and in the statement. The chance of rolling a 5 and then a 4. You can use the additive rule whenever there's an or in the situation. So let's say this time I want to know what's the chance of me rolling a 4 or a 5 on my first roll. Well, the chance of rolling a 4 is 1 out of 6, and the chance of rolling a 5 is also 1 out of 6. So if I want to know what is the chance of rolling a 4 or a 5 on my first roll, then it's 1 out of 6 plus 1 out of 6, or 2 out of 6, and if I reduce that, that's 1 out of 3. So 4 and 5 occupy 1 out of 3 of the spots on a die, and so I have a 1/3 chance of rolling a 4 or a 5. Okay, so back to genetics. If I go to a tri-hybrid cross, I say I don't want to draw out 64 squares. I just want to figure out what are the chances of a certain outcome of offspring. We're going to introduce a new trait, and that's flower color. Capital P is purple, and that is dominant over lowercase p, which is white. And then we have our familiar yellow and green, and smooth and wrinkled over here. So now we're following 3 traits, okay, all at the same time. Last time we followed 2, and we started off with 1. Now it's 3. They are both dominant, okay, both heterozygous. Both individuals are heterozygous, and I want to know the chance of purple and yellow and round, okay? So purple is dominant. The chance of being big P, or big P, little p is 3 out of 4. Okay? Remember the ratio of 3 to 1? 3 out of 4 express the dominant phenotype. So it's 3 out of 4 of these individuals are going to be purple from this cross. Now let's look at yellow. What's the chance of being yellow? Well, you can be yellow if you're big Y, or big Y, little y. It's the same situation. 3 out of 4 times these individuals will be yellow because of the 3 to 1 ratio for one trait. What's the chance you're going to be round? Well, round individuals are big S, or big S, little s. There'll be a 3 to 1 ratio of round to wrinkled. So 3 out of 4 P's from this mating will be round. So if I want to know what's the chance that they are purple and at the same time yellow and at the same time round, so there's ands in there, I would multiply 3 out of 4 times 3 out of 4 times 3 out of 4, or 27 out of 64. 27 out of 64 of those plants will have purple, yellow, round seeds by what is predicted by probability. Okay? And if I drew out a Punnett square with 64 squares in it, 27 of those squares would be purple, yellow, and round. Okay? I can do the another situation. What's the chance of being white and green? White or green or wrinkled. Well, the chance of being white is 1 out of 4 because white is recessive. 3-1 ratio, 1 out of 4 by chance will be recessive. This is also recessive, so also 1 out of 4. Wrinkled is also recessive, also 1 out of 4. What is the chance of being white or green or wrinkled? Okay, so if you're white and then you have yellow, that would count because you have one of the conditions met. If you're green and you're also purple, that would count. Just trying to find out the chance of being white or green or wrinkled if you have one of those three things happening. Okay, so this is or in there. So I add up 1 out of 4 plus 1 out of 4 plus 1 out of 4, or 3 out of 4 individuals in that Punnett square will be recessive for one of the three traits. And only 1 out of 4 won't be recessive for one out of the three traits. So you can use these rules to, instead of drawing out the 64 square, or if you can do 4 traits or 5 traits or whatever, you just use the multiplicative or additive rule depending on what you want to find out. Okay, so here's a question that I'm going to give you in H5P. And then your book sort of leaves genetics there. You're going to, if you continue in as a biology major, you're going to take a whole course on genetics. And the reason is it's much, much more complicated than what I've given you in these couple podcasts. What I've given you in these couple podcasts is what's called Mendelian genetics, named after Mendel, and or sometimes called simple dominance and recessive rules, because they're relatively simple compared to real life where things are more complex. So in real life, one gene can control another gene. So you're not talking about a gene that is either dominant or recessive, but one gene is controlling the action of another gene. So you need to know what's going on at both of those genes at the same time in order to figure out what the animal or the organism is going to look like. That's called epistasis. So in labs, their coat color and their nose, lips color, eyelids color is controlled by more than one gene, and that's how you can get parents who are, say, black labs having yellow labs, or chocolate labs, and that's called epistasis. You can have. We've only given you situations where an allele is either dominant or recessive. You can have two alleles expressed and they're both dominant at the same time. That's called incomplete dominance. This can happen in flowers, where you get two colors expressed at the same time, or they're blended. One's called codominance, one's called incomplete dominance. You can have traits controlled by dozens of alleles, not just either your purple or your white as a flower, but you can have all kinds of alleles contributing to what you're going to see as terms of phenotype. So human height is an example of these. It's not that people are either short like me or tall like LeBron, but there's all kinds of variability in between, and it's colored. It is controlled by dozens of genes. Skin color is another thing that's controlled by dozens of genes. It's not that people have either one tone of skin or another tone of skin. There's a whole gradient of tones of skin, and those are controlled by dozens of individual alleles. Traits can be linked to sex chromosomes, so that you are more or less likely to have them if you are genetically male or female. And traits can be influenced by environment, and very often are influenced by environment. This is called genetics by environment interaction. An example down here are these hydrangeas, and depending on, with the same genotype, the same genes, depending on the pH soil that they're put in, you can get different colors of flowers. So lower pHs get blues, higher pHs you get pinks, and so you're going to learn lots more about this in another class. We're just getting you going with genetics here in Principles.